increase in mass in preparation for cell division. The period during which DNA is synthesized, the DNA they duplicate, and then they prepare for another growth and proliferation. The chromosomes coil and become shorter and thicker and visible under the light microscope. The duplicated homologous chromosomes appear.
The centrioles are at opposite poles of the cell. The pairs of homologous chromosomes now as tightly coiled and condensed as they will be in meiosis. Two chromosomes of each bevelent separate and start moving toward opposite poles of the cell as a result of the action of the spindle. Notice that in anaphase 1, the sister chromatids remain attached at their centromeres and move together toward the poles. In telophase 1, the homologous chromosome pairs complete their migration to the two poles as a result of the action of the spindle.
Cells produce proteins and grow in size, getting ready for meiosis II, while centrioles duplicate and start to produce spindle fibers. Both of the cell membranes break down and dissolve. Once the membranes have dissolved, spindle fibers get hold of the chromosomes on their ends, preparing to move them to the right location. Since spindle fibers are attached to the centrioles, as the centrioles move to the opposite end of the cell, chromosomes complete the lining up. Each chromatids of a chromosome face the opposite poles. Unlike meiosis I and similar to meiosis, chromosomes line up vertically under and on top of each other instead of side to side with their homologous pair. Centrioles move away from one another, with spindle fibers holding onto each chromatid of a chromosome. The chromosomes are cut into half from the centromere, become separate chromatids, and are dragged away from each other with another chromatid from a different chromosome beside them.
4 haploid nuclei are formed. Division of the cytoplasm during cytokinesis results in 4 haploid cells. Each haploid cell contains 23 chromosomes and 1 chromatid. Process to separate the cell in half and ensure that one nucleus ends up in each daughter cell. The contractile ring shrinks at the equator of the cell, pinching the plasma membrane inward and forming what is called the cleavage furrow. Eventually, there are 2 separate cells, each bound by its own plasma membrane.